And from what we know about the mitochondria so far, what really happens inside it, and how is it relevant to your research?
I mean, mitochondria, in a time, it was really underappreciated organelle. And the only thing that people were thinking about production of ATP and bioenergetics complexes 1, 2, 3, 4, and then hemosmotic theory of proton shuttling, and that's it. that was the sole function of mitochondria, and then people didn't bother to, like in 60s, and Mitchell got a Nobel Prize for that hemosmotic theory of production of ATP. And then after that, mitochondria was kind of neglected. And then later on in, I think, probably starting in the 80s, late 80s and 90s, now all the different functions starting to appear for mitochondria, like from growing apoptosis, signaling, metabolism, then innate immunity. And now we can see that this kind of exponential growth of different papers studying mitochondrial biology, different aspects of mitochondrial biology. And yes, so as we know, the endosymbiotic theory of how mitochondria appeared, there was one eukaryotic cell with a nucleus that engulfed another bacteria, and then that bacteria became mitochondria, and then shuttled all its own genes, primordial bacterial genes, to a nucleus of the cell that engulfed the bacteria. In that sense, there's a lot of interconnection between mitochondrial organelle and then nucleus, because it's in a way it's like two organisms, one living inside another, and they have to adjust their ratios of activities, let's say, especially in mitochondria, because it produces necessary energy equivalent, as we discussed, for lots of different processes. And besides that, participates in major cellular functions like cell death, innate immunity, and like a cycle that provides a lot of different metabolites for major biochemical pathways in a cell. And so it's hard to underappreciate the role of mitochondria in any field of biology now, because wherever you look, you can look cell biology, microbiology, in immunobiology, anywhere, developmental, anywhere mitochondria will have its role. So it's so universal, and now we know so much of it. That's it's participate in a lot of different functions, which is clear. The same way as nucleus is important in terms of gene regulation, gene expression that provides all necessary information for the cell. The same way as mitochondria is important in that sense, because it provides all the necessary, let's say, currency for all these processes to happen, including gene expression that happens in nucleus.
Yeah. So as you said, mitochondria is currently or now a very prominent organelle in a lot of fields in biology, but before it was quite undiscovered and nobody really researched it in depth. So a lot of new functions and a lot of new applications are also starting to pop up. So there is research to show that defective mitochondrial DNA leads to premature aging. So is it possible that the damage to empty DNA can be prevented to slow down the process of aging or the rate of aging in the future?
Yeah, that's an interesting point. Yeah, besides aging, there are a lot of effort in connection with mitochondria and Alzheimer or Parkinson's disease, and all of different proteins that participate in that. But one thing that in these complicated diseases, or in term aging is not disease, it's a physiological process. But in those complex physiological process or diseases like aging or cancer, which people also think about using different mitochondrial markers or mitochondrial DNA numbers as a markers for different types of cancer. It's important to look at the aspect of mitochondria, but it's not the only thing, and it requires a lot more knowledge, including my knowledge of what's going on with mitochondria, to be able to understand what's really going on and what the specific patterns that we can see to be able to solve that question in terms of aging and or other diseases. Because besides mitochondria, there are all the other organelles that also can play a role, and there are also the role of nuclear gene expression. And in mitochondria, there's some questions about production of one of the theories of reactive oxygen species and other things. But besides like molecular level of understanding of this process, including aging and other diseases, it's really important in biology, and this was proposed by one of the famous evolutionary biologists, Theodosius Dobzhansky, geneticist, that every question needs to be looked at with respect to evolution. And to be able to fully answer and understand aging or other different process, we need to understand what's the evolutionary basis and what's the evolutionary reason for that specific process to happen. That's why understanding at the molecular level is really important, but we also need to ask questions, not just how it happens, but why it happens. Proximal cause, understandable, but more distal cause is also important.
Yeah. In your prior research on replication transcription in human mitochondria, it was stated, and I quote, that the interaction of the human transcription elongation factor, TEFM, with mitochondrial RNA polymerase and nascent transcript prevents the generation of replication primers and increases transcription processivity. What was the process behind reaching this conclusion?
Yeah, so human mitochondrial DNA is a really interesting subject in that it's different from the other mitochondrial DNAs by size. It's relatively small. It's like 16,000 base pairs, and it encodes about 12 protein genes and 15 ribosomal RNA genes, tRNA or ribosomal RNAs. It doesn't have any introns or non-coding sequences as nuclear genome has. And there is this enzyme that is responsible for generation of transcripts or molecules that then give rise to the proteins, right? Which is RNA polymerase. It also has another function that provides specific primers or specific sequences for in other enzymes that creates more DNA polymerase. And the reason that we were studying that, it was unclear how interaction between RNA polymerase that creates transcripts to make proteins, which one group of molecules kind of coincides with another enzyme, DNA polymerase, that is responsible for just increasing numbers of mitochondrial DNA. So what's the resolution? And then we figure out and learn about this newly identified protein, which later on was now became known as transcription elongation factor. At the time, it wasn't clear whether it was transcription factor or elongation factor. And we came up with hypothesis that it may actually help to regulate this switch or transition between one process versus another, basically between creating necessary transcripts to make proteins or versus making more DNA. And in mitochondrial DNA, there is a specific roadblock at the beginning. That's why RNA polymerase can make these primers. And basically, when it hits that roadblock, RNA polymerase, it stops. And it provides the sequencings for another enzyme that makes more DNA. And as soon as we thought that if you can prevent that roadblock, then this RNA polymerase wouldn't fall off, wouldn't stop there, and then just continue going and making it function more transcripts. And actually, it turned out to be true because with the help of another elongation factor, the protein, that prevented that roadblock, and then RNA polymerase could continue to perform its function. Okay. That was the major thing that we discovered. Yeah.